Human Embryology, Part 1. Embryo Development. Questions for the final exam. Embryology, the science about the embryo development. Embryogenesis, it's prenatal, antenatal development. Embryonal prenatal period start from fertilization and finished by birth. Its duration 280 days or 40 weeks or 9 months. It consists of 3 main periods. Preembryonic period, the first week. Embryonic, from 2nd till 8. And fetal, from 3rd till 9 months. And in the preembryonic period, new organism called preembryo or conceptus. The second called embryo, and in the fetal, fetus. To the end of the embryonic period, the embryo reach 4 centimeters in length and 5 grams weight. There are 5 stages of embryogenesis. Fertilization, several hours, finished by zygote formation. Cleavage, 6 days, and as a result the blastule formed, 1 germ layer embryo. Gastrulation, from 7th till 17th days, the gastrule formed, 3 germ layers embryo. 4th, histogenesis and organogenesis, after gastrulation, formation of primary tissues and organs. And systemogenesis, from the 2nd month till birth, formation of systems of organs. Progenesis, period of development and maturation of sex cells and organs, providing the condition for fertilization and development of the new organism. Formation of male sex cells, called spermatogenesis. It consists of 4 stages: proliferation, growth, maturation and formation, and take place in convoluted seminiferous tubules of testes. It starts following sexual maturation and finished after 60 years old, its reproductive period. The development of spermatozoa is taking place permanently, and every day produced more than 300 millions of spermatozoa. Stage of formation demonstrated here. Formation of mature spermatozoa from spermatid. During this period, the head, this acrosome, appeared, and flagella, tail. Agrosome developed from Golgi complex and structure and function like flat, large lysosome containing hydrolytic enzymes which will destroy membranes, outer membranes of oocyte. Head contain also the small elongated nucleus is very dense and contain only heterochromatin, inactive, highly spiralized. Tail consists of 4 parts. Connective piece contain 2 centrioles, proximal and distal, which produce the axonema, which consists of pair of central and 9 pairs of the peripheral microtubules. In the middle piece, around axonema, located numerous mitochondria on the spiral plane. Principal piece contain microtubules and circular fibers around, and end piece contain only axonema without fibers around. Spermatozoa have developed in testis, final maturation, and during ejaculation spermatozoa release into the urethra, mixed here the secret of the 3 accessory glands, and release outside and enter female reproductive organs. This is a picture of spermatozoa in ejaculate smear. Heads and tails visible, and sometimes acrosome. For fertilization it necessary more than 300 million spermatozoa in ejaculate, and most of them should be normal, alive and motile. Spermatozoa can move quite quickly with the speed about 50 microns per second. Ovogenesis, female sex cells formation, consists of 3 phases: proliferation, growth, small and large, and maturation. It takes place in ovary, reception of final maturation. It takes place in uterine tubes, and second division of meiosis may be completed only after or in the course of fertilization by spermatozoa. It starts before birth. Small growth take place during the life before, and in the course of sexual maturation till menopause, and large after sexual maturation only, and maturation take place just before and after ovulation. Duration during embryogenesis, first phase, during life till menopause, and large about 14 days. Maturation 1-2 days before and in the course of fertilization. So the development of female sex cells is taking place by cycles. During the sexual cycle, usually only 1 oocyte produced. So the development of oocyte takes place in the ovary, then ovulation, and oocyte release and enter the uterine tube, and slowly moving in the direction to uterus. And somewhere here in ample of oviduct, spermatozoa can find and reach and fertilize it. But before, spermatozoa should spend several hours in the uterus, and only after the activation, capacitation, they start to move actively and found and fertilize the ovum. Oocyte, after ovulation, it's the largest cell of the body. It reaches 150 micrometers in size. The nucleus, this nucleolus in the center, in cytoplasm, few yolk inclusions. Cortical granules under the cell membrane. Cell membrane is waving because oocyte microvilli. The second membrane is zona pellucida, quite thick, and contain receptors to spermatozoa on its surface. For recognition the oocyte by spermatozoa. And around there are follicular cells with their processes go through zona pellucida and reach the surface of oocyte and feed in it. Fertilization is the process of penetration of an ovum by spermatozoon and the subsequent fusion of the haploid gametes, and zygote will formed. There are two phases, distant and contact interactions. Spermatozoa create androgomons and oocyte secrete gynogomons, and they exchange distantly by information, and it helps to spermatozoa activate it. It's called capacitation, start to move actively with their flagellus, and information from spermatocyte help to maturate to oocyte prepared to fertilization. And spermatozoa can move actively and reach oocyte, find oocyte using these chemical markers, signals. And only 100 spermatozoa survive and reach the ovum, and altogether they destroy the corona radiata. They recognize oocyte through the receptors of zona pellucida, and other spermatozoa agglutinated and died. Fertilization, contact interaction. You can see the surface of the oocyte, corona radiata, and spermatozoa, which using the enzymes of the acrosome destroy corona radiata jointly. Many spermatozoa try to destroy zona pellucida, but the quickest, which reach the cytolemma or oolemma and will fertilize, head and neck enter, and tail still outside. The neck bring the centrioles necessary to finish the second division of meiosis, and quickly the mature ovum has appeared. This is acrosomal reaction. Hydrolytic enzymes of acrosome release outside and destroy the zona pellucida. So, fertilization, then 100 sperms destroyed corona radiata. Here's the first polar body after first mitotic division, and second mitotic division take place only after the entrance of spermatozoa into the oocyte. Then the second mitotic division finalized, the mature oocyte has appeared, and this is female pronucleus, and from the head of spermatozoa the male pronucleus has appeared, and they move close to one another, and this figure called syncaryon, and then nuclei fuse, and new cell, this diploid set of chromosome has appeared. It zygote, the new organism and the face of one cell. Then second stage, cleavage, duration 6 days, take place in uterine tubes 3-4 days, and in the uterus lumen 2-3 days. It's full in regular asynchronic division. Initially 2 daughter cells called blastomers appeared, then 3, 4, 5, more and more. 16 cell stage, morula, the 3rd day of embryogenesis. The cleavage, some mitotic divisions, but the daughter cells had no chance to increase in size before next division, because the interface too short, only for reduplication of DNA before next division. They called blastomers. After 4th day of embryogenesis in the uterus lumen, blastomers continue to proliferate, dividing, and start to accumulate the fluid from uterus lumen. These small cells, one-layer cells called trophoblast. Here cell mass called embryoblast, and blastocell, the cavity. From the 7th day, blastocysts start to implant into endometrium, and this phase of menstrual cycle, predmenstrual or secretory phase, when the endometrium prepared for implantation. This is the embryo. Implantation duration about 40 hours, consists of 2 phases: adhesion, then embryo glue, the surface epithelium, and invasion, and when cytotrophoblast converted to plasmodia syncytia or symplastotrophoblast. And these primarily release hydrolytic enzymes which destroy epithelium, connective tissue of the lamina propria, and finally the blood vessels to get the necessary food components directly from blood. So implantation, adhesion, invasion into the endometrium, growing and became implanted deeper and deeper, quite quick, the epithelium lining the endometrium, growing and close the defect. This is an embryo inside the uterine wall, endometrium, after the implantation completion. Epithelium recovered, so from the surface implanted embryo not visible. The cleavage and pre-embryo move in the direction of the uterus. It implanted in the uterine wall, became bigger and bigger. As far implantation start, the input of enriched food components enter the embryo, it can continue the development. The first phase of gastrulation takes place on the 7th day of embryogenesis, and it's resulted in the formation of 2 germ layers by delamination, epiblast and hypoblast. Then the development of the embryo body itself stops before the provisional organ has appeared. There are 3 main provisional organs: amnion, then yolk sac, and then from trophoblast, site and plasmodial trophoblast, developed the chorion by red color underline extraembryonic mesoderm here and here around the extraembryonic membranes. This slide demonstrates the 7th day of implantation and the beginning of gastrulation. Then embryonic disc divided into epiblast and hypoblast. After the formation of extraembryonic membrane, the second phase of gastrulation takes place, the formation of the 3rd germ layer. You see the embryonic disc, epiblast and hypoblast, and in the beginning the cells of epiblast start to proliferate intensively and move to the caudal direction, and then the cellular streams fuse and start to enter the another direction. The thicker part of epiblast called primitive strake, and this round shape thicker part called primitive knot. Then here appeared the invagination and cleft, and through this hole the cells of the epiblast migrate under epiblast, between epiblast and hypoblast, and form the new layer, mesoderm. At the same time, cells of epiblast push and remove the hypoblast and form the embryonic endoderm. Here the remnants of epiblast form ectoderm and between mesoderm. And this is microphoto from your histological preparation, primitive strike. Some thicker part and invagination. Axial organs formation take place after gastrulation. In the middle of the primitive strake, the nerve plate appeared. It's invaginated in the middle part, continue, and from the anterior thicker part of the nerve tube appeared the brain, and from the body's part have appeared the spinal cord. And here the group of cells migrate from side part of neural groove and compose the neural crest. This is material of peripheral nerve ganglia. Embryo 3 germ layers. It's the microphotogram from your histological preparation. It demonstrates the embryo in the age of 24 days. There is ectoderm, endoderm, and mesoderm. Mesoderm dividing into 3 parts: Somits, segmented part, segmented legs, and splanchnotomes, visceral and parietal layer. So ectoderm, endoderm, nerve tube, and chord. And the slide demonstrates also the beginning of folding, then side parts of the embryo invaginated in this direction, and folds appeared, they fused together, and flat embryo converted to 3D shape embryo to the end of the first months of embryogenesis. There are 3 germ layers: ectoderm is blue, endoderm it's yellow, and mesoderm between. It consists of segmented part, segmented legs or nephrogenotomes, and splanchnotomes, visceral and parietal layer, this celome between. Fourth stage, histogenesis and organogenesis, the formation of tissue and organs from 3 germ layers and 3 axial organs. Differentiation of ectoderm. The nerve tube and nerve crest appeared, and brain and spinal cord, as well as eye retina appeared from nerve tube. Nerve crest also the source for chromaffin tissue, and skin ectoderm is the source for epidermis development and its derivatives. It also compose the epithelium, vagina, and rectum. Differentiation of endoderm, epithelium and gland of the stomach and intestine, epithelium of liver and pancreas, and in addition the ectoderm and endoderm jointly form a precordial plate. It's the source to the epithelium of esophagus, bronchi, thymus, thyroid and parathyroid glands. Differentiation of mesoderm, initially for 3 parts: somits, nephrogenotomes, and splanchnotomes. And in somit, the surface part differentiated to dermatomes, the source of derm. Sclerotome provides the material for cartilage and bone formation, and middle part called myotome because it's the source of the striated skeletal muscle. Nephrogenotomes, the source for development of kidney and reproductive system. Splanchnotom, mesenchyme, provide microglia and smooth muscle cells. A visceral layer of splanchnotom form the myocardium and adrenal cortex, and parietal layer provide the mesothelium for all 3 serous membranes. This slide demonstrates the embryo development. Embryo inside the uterus. This picture demonstrates the preembryonic and embryonic periods. So embryo development during 2 months. Initially the embryo is very small, that in growing, and from 30 days it has a specific shape with small head and long tail. Then the head became larger, it's growing, and tail will disappear. And to the end of the embryonal period, embryo reach 4 centimeters in size and 5 grams in weight. It's the development in fetal period, the fetuses. And this is a newborn child. And it's the final result of embryogenesis.